Okay, so here we go. Our first notes from modified biology. So this week, what we're going to be looking at is something that you guys have probably looked at every year that you've taken some sort of science class. And what we're looking at here is the scientific method and the steps of the scientific method. So when you think about the scientific method, all it means is a way to solve or how to solve a problem. So you come across problems every day in your life. Maybe you walk into your bedroom and you flip the light switch and the lights don't come on. That's a problem. You think about it, but you don't actually go through the steps of the scientific method in order to solve that particular problem of getting the lights back on. So maybe you just go, oh, well, the lights are out, I need a new light bulb. Well, you really do kind of go through each one of these steps, whether you realize it or not. And these are the steps that we're going to follow in order to go through different experiments within class. So anytime that we are given an experiment, you guys will always be presented with some sort of problem. You need to know, why are we doing the experiment? So it's a statement that you want to solve. Along the way, you need to know have some sort of research, either class notes that you have, information from your past experiences, a textbook online, maybe you're going to ask a friend, maybe you look at an owner's manual. So if you're thinking about that light bulb problem, you research it because maybe you've changed a light bulb before. Maybe you've hit a light switch and the lights don't come on and mom or dad told you to go get a new light bulb and put the new light bulb in. You knew that it worked. So all of that is research. It's basically a way that you are thinking about how to solve that particular problem. Once you have that problem and you've researched the information a little bit, you are going to create a hypothesis. Now, hypothesis is an important part of an experiment. And a hypothesis is an educated guess. Say it again. It's an educated guess, which means it's a possible solution to the problem. It does not have to be correct. So don't go through and go, oh, my hypothesis is wrong. She's going to take off points for that. Let me go back and fix that and put the right hypothesis in. Absolutely not. You will never lose points for having a wrong hypothesis. You'll explain it more or less in your conclusion. I have no problem. It's an educated guess based on research information on how to solve that problem. Doesn't mean you're right. Maybe you go and get that light bulb and you put the light bulb in and guess what? The lights still don't work. Maybe it was a fuse and then you're going to go back and try that again. So just because you have an educated guess doesn't mean it's always going to be correct. It's one of the ways that we're running through this particular experiment. What I will mark off for on your hypothesis is whether or not you are bold with this. So there really should be no personal pronouns anywhere within a write-up or any of these steps of the scientific method. So you should never really put, I think or I believe. So in other words, I think the lights don't work because the light bulb is blown. You don't want to be wishy-washy on that. So no, I think. I believe the lights went out because the light bulb didn't work. Nah, still kind of wishy-washy. The lights went out because the light bulb was blown. The lights went out because the fuse was blown. Whatever it may be, get rid of I think, get rid of I believe. There should be none of that. Start with your statement, be bold with it. And that's the only place that I'm going to be taking off points for. Okay, with your hypothesis, it should be a because statement. The lights were blown because, or the lights did not come on because the light bulb was blown. There you go. So make it kind of a because statement. Once you have that hypothesis or educated guess, that's where we're going to experiment. If you were home with the light bulb, you go get a new light bulb, you put that new light bulb in, you go over and hit the light switch. That was your experiment. Didn't work, you go back, create a new hypothesis. The lights don't come on because the fuse is blown. Go experiment. Go to the fuse box, click the fuse over, go back, turn it on, boom, solved. So your experiment is basically doing it. You're testing that hypothesis. In class, when we do that, we're all going to be collecting some data. And what you're going to do is you're going to be putting that data into a data chart, possibly graphing that particular data to give us a better view of the information. And that will allow us to come to our last step after experiment. We're going to go through conclusion. We're going to analyze the data that we collected during our experiment. So what did we learn? Was our hypothesis correct? Explain what happened and why. So we're utilizing the information that we had seen previously, and now we're going to go through and kind of solve it. Okay, think about that data and analyze it. What happened and why? The hypothesis was correct. This happened. The hypothesis was not correct. This is why. I don't care if your hypothesis is correct. Explain to me what happened and why that hypothesis is wrong. If that hypothesis was not correct, had this been an actual experiment, you would actually come up with a new hypothesis and go through and test it once again. Okay, so we would keep doing it, just like that light experiment. So lights are out, I go and I experiment, I change the light bulb, I go hit the light switch, they still don't come on. The hypothesis was not correct, the lights did not come on after the light bulb was changed. The new hypothesis is that the lights will not come on because the fuse is blown. And then I'm going to go back and I'm going to experiment again. So once again, my steps of the scientific method in order is problem, research, hypothesis, experiment, and conclusion. So when we take those particular steps and we put them into a lab or an experiment, there's some other things that we need to think of. So when we're setting up an experiment, within that experiment, we're going to have what are known as a control and an experimental or variable. Your control is the normal. What do we normally have? What do we normally do? That's how we set up our first experiment. Our second experiment is basically thinking about our hypothesis, what are we changing, and to test that particular hypothesis or experiment. How is it different than our normal? So the variable is going to be that experimental group. Your control is going to be used for comparison. Once again, it's our normal. Our variable is the one thing that is being tested in the experiment. It's the one thing that changes in the experiment. It's the one thing that you control in that experiment. And we're going to be doing some experiments along the way, and we'll be going through different examples of controls and variables. Your variables are what are going to be going onto your graph. So within those variables, you have what is known as an independent variable and a dependent variable. So if you've ever set up a graph, we have our x-axis, which is horizontal, and we have our y-axis, the up and down axis. Your x-axis is known as your independent axis. That's that horizontal axis. This is what you control in the experiment. This is the variable that we're going to see. So maybe you want to check growth of plants every 5 days. I can control how often I'm going to check the height of those plants. I'm going to say every 5 days I want to look at the height. Day 5, day 10, day 15. What I cannot control is known as my dependent variable. That's going to go along the y-axis. I cannot control the results of the experiment. I cannot control how high those plants are going to grow. So every 5 days that I'm checking them, which is what I do control, and that goes along the x-axis, I do not control the height of them. That will go along the y-axis. As I put this information on my graph, I want to make sure that I'm including units. So along the bottom, I can't just put 0, 5, 10, 15, 20, 25, 30. Well, what do those numbers represent? I'm going to put days. I want to make sure people know it's not hours or minutes or degrees Celsius for that matter. Along the y-axis, what am I measuring in? Am I measuring centimeters, inches, meters, feet, miles, days, temperatures, whatever it may be? You need to let people know what you're measuring in here. So always include your units. Within your experiment, you guys are going to see constants. Notice it's plural. You will have many of them. Everything that stays the same between your experimental groups and your control group. So if I'm growing those particular plants that I want to measure, and I'm measuring them every 5 days, those plants need to be grown in the same type of pot, same size of pot, same type of soil. They should be the same type of plant. I should be measuring the same way. They should have the same amount of sunlight. Those are the constants. What's the same between each one of my groups? Because the only thing that's going to change is my variable, the one thing I'm testing. And maybe what I'm testing is plant A gets 5 drops of water each day. Plant B gets 10 drops of water each day. Plant C gets 15 drops of water each day. So maybe I'm just testing the amount of water that they're given. Everything else should be the same. I can't say plant A gets 5 drops of water and is put in sunlight. Plant B is given 10 drops of water and put in no sunlight. Because then I don't know what makes it grow faster. Is it the water or the sunlight? Because I'm testing 2 different things. I only want to keep it to 1. So those are our constants, or things that stay the same. And once again, it's going to be multiples. You can't just give me one constant. You have to give me multiple numbers of constants that stay the same. So from that, we're collecting our data. This is the information that we basically collect and record, or gather and record from the experiment. We are going to create data charts, and a data chart just basically helps us to organize the information so whoever may be looking at our experiment can go, oh, there's our data. Everything's nice, neatly organized, and it's labeled. You guys are going to be looking for two types of data, and think about the words with these. So we have what is known as qualitative data and quantitative data. So they look alike but break the words down. So I'm going to start with the second type, quantity. You guys probably have heard that word before. How much of quantity of something do you have? So quantity is talking about numbers. It's actual measurements, actual numbers. So the plants are growing 5 centimeters. The plant grew 7 centimeters. The plant grew 9 centimeters. The temperature was 45 degrees Celsius. It was 50 degrees Celsius. This is usually what we're going to be putting onto our graphs. So quantitative data is data that is actual numbers, actual measurements. Qualitative, think about quality, is more descriptive. It is words. It could be color. It could be height. So if we're talking about those plants again, maybe one plant's small, maybe one plant is medium, maybe one plant is large. Maybe the leaves on one plant are dark green. One plant has medium green. One plant kind of has yellow leaves to it. Maybe it's a smell something has. Maybe it's a texture something has. The clouds look fluffy. The tree is tall. The sidewalk is long. That's all qualitative data. Okay. Next, once we have all that data, we are then going to go through and analyze the data. We want to look at our results. We want to draw a conclusion. Was our hypothesis correct? Yes or no? Yes, the hypothesis was correct because this is what my data shows. This is what my data is telling me. And kind of include numbers with that when we're going through our conclusion. If your conclusion supports your hypothesis, you're done. Yep, the hypothesis is correct. My data tells me that this is exactly what my hypothesis said. We're good. If you're like, oh, no, my hypothesis was wrong, you need to go back and kind of start it again. You need to create a new hypothesis. You would change the experiment to fit that hypothesis, and you would go through and collect new data. Along the way, we're going to be doing some little biome activities, little pond water activities, and collecting some qualitative data. In order to do that, we need to use observation skills. So when we observe something, it means that you are using senses to collect data. So usually things that we don't use is taste within class. I don't want you guys tasting anything. Oh, this tastes sweet, tastes sour. We're not doing the tasting thing. But we can use textures, we can use smells, we can use sounds. Okay, so think about all those different senses that we can utilize in order to collect your data. And that's going to be one of the things that's going to help us come up with our hypothesis along the way. Okay, so think about those steps: problem, research, hypothesis, experiment, and conclusion, in order to help you with those steps of the scientific method. These notes will help you guys go through a little scenario problem that you guys will be doing on your own. That will be the next video for you. So this is the end of your scientific method notes. Go back, rewatch that video if you need to. This note sheet itself will be downloaded onto Google Classroom, so that this is just there for you guys to go back and review at any time. So it will be listed under Scientific Method under the Classwork tab. When you open up that Classwork tab, on the left-hand side will be a list of topics. You'll click on Scientific Method, and then you should be able to pull up those particular notes. Let me know if you guys have any questions on this information.